I have no conflict of interest to disclose related to these contents of my talk today. So all of you know, and I don't have to spend too much time on that, puberty is a fascinating developmental process, and it's also quite multifaceted. So different developmental events are taking place in order to define what we understand as puberty. And I would like to stress that the interest of my laboratory is mostly related with trying to understand how during puberty the HPG axis, or the reproductive axis, becomes fully activated. And also, as indicated in the scheme, how metabolic factors coming from different tissues, such as, for example, the white adipose tissue, is controlling or modulating this complex phenomenon. In this, we have been studying different mechanisms controlling the activation of GnRH neurons located in the basal forebrain, and I will tell you more about this during my presentation today. In the particular context of puberty, the activation of GnRH neurons is absolutely critical and is driven by a change in the balance between excitatory and inhibitory inputs coming down to these GnRH neurons. And as I said before, much of the efforts of my laboratory have been focused in trying to understand how this balance is changing during pubertal maturation, and actually how the dialogue between epithalamic signals, peripheral factors, and environmental signals, including nutritional cues, is taking place to define puberty to occur in a normal time or to occur earlier or later than expected. And indeed, in dealing with that, actually we have been focusing on some of the key words, if you want, that I will be using in my presentations, such as dyspeptin, puberty, obesity, and nutrition, and I will illustrate you on that in later slides in my talk. Why we are interested in puberty and the nutritional control of puberty? Well, indeed, first of all, we are physiologists, so we try to understand this fascinating phenomenon from a physiological perspective, but in addition to that, we are biomedical researchers, and we are all aware that indeed there are trends for potential changes of the age of puberty or puberty onset that might be related with nutritional and metabolic situations, and indeed an association between the increasing trends for obesity seen in the US and Europe are reciprocally related with a trend for earlier puberty onset both in America and Europe. So indeed, there seems to be a strong circumstantial evidence suggesting that in fact, obesity and other metabolic problems might be behind some of the pubertal or alterations of puberty that we are seeing these days. And in fact, these trends for early puberty, whose mechanisms remain largely unknown, as I said before, metabolic factors might be important contributors, but also environmental factors and endocrine disruptors have been blamed for some of the trends that have been reported in the literature that appear to be sexually dimorphic in a way that girls are certainly more affected by these trends of earlier puberty, but now there are appearing more and more reports suggesting that in fact boys could be also facing this earlier entry of puberty, at least in some series, especially in the context of obesity. And the aspect that fascinates us the most is that in fact these changes in the age of puberty could be important not only because affect this fundamental developmental phenomenon, but also because somehow increase the risk of having more serious diseases later in life, including a wide number of adverse health events such as cardiovascular or metabolic diseases, higher risk of some types of cancer, of course perturbation of adult health, and also psychological alterations. And I show you here one particular example of a paper coming out from Ken On's group in Cambridge showing that indeed 48 different adverse health outcomes can be related with either precocious or late puberty, both in girls and boys. So indeed, we believe that it's important to try to understand how puberty is controlled, why puberty is being altered, what are the factors, and eventually how this could be prevented in the human population. So just as it has been shown or proposed for obesity, we believe that to a large extent, many of the alterations of puberty are centered in the brain or occurring through alterations in pathways, of brain pathways. And in fact, we are very much focused or interested in trying to understand how different hypothalamic pathways converging onto GnRH neurons are contributing to the normal control of puberty and its deviation in pathological conditions. So In studying those hypothalamic mechanisms, and to put it in a very simple manner, I could say that most of what we have been doing in the last few years in the lab is related with a kiss, but it's not the sort of kiss that you would think about when you are in Vienna, but rather another less artistic but very exciting kiss, which is the KISS1 system. I show you here the elements of this system in which the gene is called KISS1, and is encoding a family of proteins called kispeptins that are acting through the GPR54 receptor, or also known as KISS1R. And actually, this KISS1 system was initially related with basically tumor control, but it was in late 2003 when seminal contributions, especially from Nicolas Roux here in the audience and other groups in UK and US, somehow put the KISS1 system into the reproductive scene by showing that, in fact, inactivating mutations in the receptor for kispeptin, GPR54, is causing central hypogonadism, so a failure of the reproductive axis to become activated at puberty and fully functional later in life. And actually, in just about 15 years, we have witnessed kind of an explosion of the kispeptin studies that have allowed the community, the scientific community, to really define what are the characteristics of the system. So now we know that there are specific neuronal populations in specific areas of the hypothalamus, such as the arcuate nucleus and the rostral hypothalamus, that engage in the control of GnRH neurons to activate GnRH neurons and indeed participate in the regulation of the reproductive function throughout the lifespan. And particularly in the context of puberty, we do believe kispeptins are playing a very important role in defining the proper timing of puberty. And to convince you on that, I have plotted here in a schematic manner some of the data that we had generated using animal models. Basically, we use preclinical models to address the function of neuropeptide pathways in the control of puberty. And just to put it in a very simple manner, I show you here one external phenotypic marker of puberty, which is vaginal opening. So in control rats, these are female rats, basically vaginal opening indicates activation of the axis, so puberty onset, and take place in a progressive manner between postnatal day 32 to 35 approximately. If we look at what happens in the brain regarding the activity of the KISS1 system, also to put it in a very simple manner, what we witness is an increase in the kispeptin tone from low levels before puberty to high levels after or during puberty. We have been able to pharmacologically manipulate the system, so if we advance this enhancement of the kispeptin tone, what we see is an advancement of vaginal opening associated with other markers of earlier entry of puberty. On the contrast, if we block specifically kispeptin actions by using a specific antagonist specifically in the brain, we get the opposite phenotype, delay of puberty onset, and also delay or reduction of other markers of pubertal activation. In other words, we do believe kispeptins are very important in defining the timing of puberty, and even though the data in humans is not so compelling, evidence suggests that this might be also the case in other mammals, including humans. What about the metabolic control of the reproductive axis, and specifically the metabolic control of puberty? Well, here the situation is a little bit more contentious because there is a strong evidence suggesting that, in fact, conditions of metabolic stress, defined, for example, by low leptin levels, are associated with suppression of kispeptin tone in the brain, and this probably leads to suppression of the reproductive axis, such as situations seen in girls with anorexia nervosa. What about obesity at the other side of the spectrum? Well, situation is not so clear, as I said, because here we have excess of leptin, and actually this excess of leptin, according to experimental animal models, including our own data, tend to activate the KISS1 system earlier. But if the obesity insult persists over the time, what you see is a kind of a paradoxical suppression of the kispeptin tone, indicating that this is probably acting in a biphasic model, meaning that at the initial stage of obesity, we have a precocious activation of the KISS1 system, whereas if it continues over the time, this leads to a suppression of the KISS1 activity at the hypothalamic level. In fact, there has been a lot of studies suggesting or exploring how this leptin-kispeptin interaction is taking place, and the situation nowadays is kind of a mixture of different possibilities, including potential direct actions of kisspeptin, of leptin on KIS1 neurons that probably are not dominant, and very, very likely leptin and other metabolic signals are acting indirectly on KIS1 neurons, as indicated also in the scheme here. So if we sum up what I have told you so far, we can kind of come to this sort of equation in which we believe that metabolic signals, including leptin, operate by modulating kisspeptin activity that in turn control G-enriched neurons, and this leads to modulation of the gonadotropic output, and this is critical for pubertal onset. However, we also think that this is not so simple, and that's why in our laboratory in the last few years we have tried to better understand what could be the molecular mediators and sensors involved in this kind of pathway that is represented in the upper part of the slide. So in this search for molecular mediators of leptin actions on the elements of the brain controlling puberty onset, we have become interested initially in two quite universal cellular energy sensors, which are mTOR and AMP kinase. And the interesting thing here is that these two sensors operate in a reciprocal manner. So conditions of energy excess, defined by excess of leptin, are activating mTOR and at the same time are inhibiting AMP kinase. And we have done a number of studies in which we have evaluated the potential interplay between AMP kinase and mTOR in regulating the KIS1 system as potential transducer of the actions of different nutritional factors, metabolic hormones, and sex hormones in the control of puberty onset. And to make a long story short, our experimental data produced over the last few years tend to suggest that in conditions of energy sufficiency, there is a predominance of the activity of mTOR in critical hypothalamic areas that leads to increased KIS1 gene expression and pubertal progression, whereas on the opposite, energy deficiency leads to a predominant activation of AMPK in line with the reported action of this cellular energy sensor, which is known to be activated in conditions of energy insufficiency, and these cause repression of KIS1 and pubertal delay. However, this tandem does not explain fully, for example, how puberty is affected in obesity conditions, and that's why we try to get deeper into the knowledge on how other cellular sensors or cellular energy sensors could interact with this tandem in controlling puberty onset. And in this search, we became very much interested in trying to define the potential roles of a family of molecules, and particularly which is called SIR-twins, and particularly one member of the family, SIRT1, which is the ortholog of the C. elegans SIR21 that has been involved in the control of lifespan, and also is known to have very critical and diverse function in the control of mammalian physiology. So all of our cells are equipped with SIRT1, and this SIRT1 molecule is important in controlling a wide diversity of functions, including metabolism and probably lifespan or health aging. And other reason why we became so much interested in this SIRT1 pathway as putative controller of puberty is not because it's acting on different tissues, but also because it has been shown to collaborate or cooperate with AMPK in mediating many of its metabolic actions. In other words, conditions in which SIRT1 is activated are also activating AMP kinase and vice versa. So we thought maybe SIRT1 is cooperating with AMP kinase in controlling puberty onset. And we did a number of studies, experimental studies, using preclinical animal models, basically female rats and female mice, in order to address this potential function from a mechanistic standpoint. So first observation we did was that during normal puberty in the female rats, there is a switch between the ratio, if you want, between SIRT1 and KIS1. So basically what happens is that SIRT1 is going down in the hypothalamus of maturing female rats, and this is associated with the well-known increase in KIS1 gene expression, being KIS1 a major driver of puberty, as I told you before. What about conditions of metabolic stress? So basically, first we applied to a well-known model of pubertal delay due to 30% subnutrition in female rats. And to make a long story short, as you can see here, this stress not only caused decrease in body weight and delayed in vaginal opening as an external sign of puberty, but also a reversal in the normal ratio between SIRT1 and KIS1. So opposite to what happened during normal puberty, SIRT1 goes up, and KIS-1 goes down, and we believe this could be mechanistically important to explain why puberty is delayed in this particular condition of subnutrition. To provide further mechanistic evidence for that, we went to pharmacological experiments in which, without touching body weight, activated SIRT1 from a pharmacological perspective, and for that we use central brain infusion of a pharmacological agonist or activator of SIRT1. What we observed here was that without changing body weight, just by activating SIRT1 activity, we could delay puberty and also the puberty onset, monitored by vaginal opening, and also delayed follicular maturations in the ovaries, indicating that indeed activation of SIRT1 at the brain level can be sufficient to delay puberty in keeping with our original observations in models of metabolic stress. But of course, as I said before, we were very much interested in trying to understand how obesity can cause alterations of pubertal timing, and for that we moved to another model, in this case of overnutrition, that is generated by combination of two obesogenic insults. First, overnutrition during lactation, followed by high-fat diet. So in this model, that I will show you here and afterwards in later slides, you get an increase in body weight during puberty, and this is associated in this case with an advancement of all signs of puberty. In other words, obese female rats enter puberty earlier, just as it has been reported for obese girls. In this particular model, what we observed, again in keeping with the hypothesis, is that SIRT1 expression or content in the hypothalamus is going down, too early, and this is associated with an increase in KIS-1 gene expression. Again, this increase in KIS-1 being possibly the driver for the advancement of puberty onset seen in this particular model. In collaboration with Sergio Ojeda and Alejandro Lomnitsky in the US, we were able to demonstrate that indeed KIS-1 neurons co-express SIRT1, and when we overexpress SIRT1 specifically in the arcuate nucleus where this prominent population of KIS-1 neurons is located, again, we were able to delay puberty, as shown here, by delay of vaginal opening and also by delay in the first ovulation. In other words, in conditions in which you enhance SIRT1 tone in the hypothalamus and specifically in regions enriched of KIS-1 neurons, you get delayed puberty. In situations in which SIRT1 is going down too early, you get earlier puberty. Together with Sergio Ojeda and Alejandro Lomnitsky, we have been exploring the molecular mechanism, and in order to keep on time, I will just show you kind of summary cartoon of what we think is happening in the brain of these models of normal puberty, earlier puberty, or delayed puberty. So during normal puberty, what we see is that there is a strong binding of SIRT1 together with other repressors to the promoter of KIS-1 in juvenile animals. And this moves into a situation in which these repressors are removed from the KIS-1 promoter during the pubertal transition. And this allows the increase in KIS-1 gene expression and drives a puberty onset. In the models of obesity in which puberty is advanced, what we see is that at an age in which this strong repressor machinery is still bound to the KIS-1 promoter in normal animals, this is no longer true in the obese animals. So you get an earlier removal of the repressor that allows increase of KIS-1 at an early age and earlier entry of puberty. On the contrast, in the model of undernutrition that is associated with pubertal delay, what you see is that the repressive machinery is still there at the expected time of puberty, and this is causing suppression of KIS-1 gene and is driving pubertal delay. So altogether, I think that when we put all this evidence together, focusing on the KIS-1, we are a little bit KIS-1 obsessed, I have to admit, but we have been able to somehow fill the gap between changes in nutritional and metabolic cues and changes in KIS-1 by showing that a number of cellular energy sensors and other metabolic mediators, including mTOR, AMP kinase, and SIRT1, are interacting as an interface between these nutritional factors and the KIS-1 system, either by activating or inactivating the system and by reciprocally regulating each other in order to define the proper timing of puberty and its modulation in conditions of nutritional or metabolic stress. But the final part of my talk I would like to dedicate to some related line of research in my laboratory, which try to address not what is upstream KIS1, but rather what is downstream KIS1. In other words, how these changes in KIS1 are transduced into changes in pubertal onset. So we are trying to understand how downstream effectors are operating and transmitting the information from KIS1 neurons to GnRH neurons and other pathways leading to pubertal onset. And just to illustrate to you, because we have several research lines running in this kind of context, I will show you some of the unpublished data that we are generating in which brain ceramides seems to be an important element of these complementary pathways connecting nutrition, obesity, and altered puberty. So ceramides is these are a large family of lipid mediators. I show you here the complex scheme showing how ceramides are synthesized. I would like to focus your attention in the upper part because this is the canonical and most important pathway for ceramide biosynthesis. And here you have first rate-limiting enzyme, cerulopalmitoyl palmitoyl transferase, or SPT, that is critically important in defining the production of ceramides in different tissues, including the brain. One of course, the beauty of this, or the importance of ceramides is well defined. They are involved in mediating quite numerous body functions, including for sure proliferation, differentiation, cell death, and apoptosis. And more recently, as I will tell you in a minute, they have been involved in mediating the pathophysiological actions of several metabolic signals. Interestingly, this pathway can be pharmacologically manipulated either by activating the system. You can feed the system with precursors of ceramides, getting increased ceramide synthesis in the tissue of interest, or you can block the de novo synthesis of ceramide with a specific antagonist. And we have made use of these agonists or activators and blockers in order to address whether ceramide signaling in the brain could have a role in pubertal control. So as I said before, it's not only that ceramides are cellular mediators for a wide variety of function, it's also that it seems that changes in brain ceramide contents are important for mediating the whole body actions of very critical or crucial metabolic hormones, including leptin and ghrelin. And even more interesting for us, it has been shown that when these ceramide levels are altered, you can see pathophysiological alterations of these pathways leading, for example, to obesity or insulin resistance. So in other words, it seems that central ceramide signaling seems to be playing a very, very crucial whole body regulatory role in the control of metabolic homeostasis, and we interrogate whether this could be also involved in the metabolic control of the HPG axis. Indeed, it was not shown before that ceramides was involved in the pubertal control, neither that it could participate in the metabolic control of puberty. So for addressing that, first of all, we come back to our model of obesity that I showed you before, defined by two combinations of insults. First, overnutrition during lactation by breeding the animals in small litters, followed by high-fat diet. And by doing these combinations of insults, of course, you get an increase in body weight, and this increase in body weight is associated with an advancement of vaginal opening, so puberty onset is occurring earlier, and also an advancement of the first ovulation, in keeping with the model that we wanted to create of obesity-induced earlier puberty. First thing we did was measure total ceramide content in the hypothalamus of those obese female rats undergoing puberty too early, and in fact there was about 20% increase in the total ceramide content. And when we analyzed the specific or individual ceramide species, we indeed observed that all but one were significantly increased in the brains or the hypothalami of these obese female rats. Okay, so this was kind of an interesting starting point. What happens if we manipulate ceramide signaling in the brain in terms of pubertal timing? Because as I said before, nothing was known about the potential role of ceramide in pubertal control. Again, we did functional experiments, taking advantage of these pharmacological agents that I showed you before. So first experiment we did was to infuse into the brain the ceramide precursor to increase ceramide content in the brain. So you see here the body weight in control animals infused with vehicle. The animals infused with ceramide precursors had no change in body weight. But when we look at the vaginal opening, we see that vaginal opening is happening earlier in these animals, and this is associated with earlier follicular maturation and earlier ovulation, indicating that excess of ceramide in the brain, as seen in obesity, can cause precocious puberty. And actually, the opposite is true for the blockade of ceramide signaling by using this antagonist or blocker, myosin, that cause no change in body weight, but delayed in vaginal opening and clear delay in follicular maturation and ovulation, indicating that you can basically mimic what happened in conditions of obesity or malnutrition by changing ceramide content in the brain through a pharmacological approach. So we, as I said before, we are a little bit obsessed with kisspeptin, so we decided to turn into the potential interaction between ceramides and kisspeptins, mostly coming from the arcuate nucleus, but also the rostral hypothalami. The first experiment we did was a physiological experiment in which we basically caused a depletion of endogenous kisspeptin by causing subnutrition to a female rat before puberty and infused kisspeptin into the brain. And we had done this before and published this before. So you see here the vaginal opening of the control animals going nicely through puberty. In those animals in which you have caused this depletion of endogenous kisspeptin by subnutrition to 25%, puberty is clearly delayed. If you administer kisspeptin to the animals without any change in body weight, so body weight is low, but still at least 60% of the animal undergo at least vaginal opening. And if you do the same, but you block at the same time ceramide signaling or synthesis, you can prevent this action. In other words, what we see here is that apparently you need a proper ceramide tone in order to have the permissive or positive actions of kisspeptin in terms of pubertal progression. So this somehow led us to evaluate whether ceramides are downstream kisspeptin to activate the classical organotropic system, which is the major action of kisspeptin known so far. And to make a long story short, we did in vitro and in vivo experiments in which we evaluated whether the stimulatory actions of kisspeptin, either on GnRH secretion in vitro or in vivo, using here LH as a surrogate markers, were affected by depletion of ceramides. And basically, to make a long story short, we came to the conclusion that particularly on these outputs, ceramides are totally dispensable. In other words, there seems to be an alternative pathway whereby this interaction is taking place. To make a long story short, we have obtained evidence suggesting that this alternative pathway could be related with another hypothalamic nuclei, in this case, the paraventricular nuclei or nucleus, in which, interestingly, we have been able to demonstrate that this nucleus receive an intense density of kisspeptin fibers during pubertal maturation. And very interestingly, in obese conditions, this density changed in an opposite manner to what we initially expected, because it goes down, but we believe this is probably related with the fact that kisspeptin are being released more actively in obese individuals. Even more exciting for us was the demonstration that this population of PBN neurons express this key enzyme for the production of ceramides, shown weakly here, and in obese individual, this goes up by more than 10-fold. So in other words, in conditions of obesity leading to early entry of puberty, you see a change in these two elements, ceramide signaling and kisspeptin pathway. We have obtained evidence that indeed the sympathetic output, because the PBN is the origin of the sympathetic innervation of the ovary, we have indeed obtained evidence that in obese female rats undergoing puberty too early, the activation of the sympathetic innervation of the ovary is increased by different markers, noradrenaline content of some metabolites, and also some molecular markers of sympathetic activity in the ovaries. So in fact, what apparently is happening here is that the increase in the sympathetic tone that goes to the ovary during normal puberty is significantly advanced in obese individuals. We have manipulated now the system by using pharmacological agents as the ones that I showed you before. And to make a long story short, we have seen that if we block ceramide signaling, we can partially prevent the early entry of puberty and also prevent or alter some molecular markers of sympathetic activity in the ovaries. And finally, just to provide direct evidence for the importance of this PBN originating system, we have gone to a specific phylogenetic experiments in which we have specifically knocked down the synthetic enzyme that I showed you before, selenium palmitoyltransferase, specifically in the PBN in obese individuals. And what we have seen is that this early entry of puberty, defined by early vaginal opening, can be totally prevented if you block ceramide signaling in the brain, specifically in the PBN, and the same happens for the first ovulation. So we altogether believe that this ceramide signaling system originating from the PBN is affecting the sympathetic output to the ovary to cause early entry of puberty in this model of obesity. So just some take-home messages to finish up. First, I hope I have convinced you, probably I didn't need to put much effort on that metabolism and reproduction are tightly connected, so changes in body weight and obesity are causing alterations of puberty, and we believe that the hypothalamic KIS1 system is an important player to connect metabolism and puberty. In our search for potential molecular mechanisms to explain this phenomenon, we have come across the important role of several cellular energy sensors, including mTOR, AMP kinase, and more recently SIRT1, that we do believe can explain how obesity is affecting puberty in mammals, including humans. And finally, central mediators such as ceramide might be playing an important role also, at least according to our preclinical data, to explain how puberty is causing early entry of puberty in our preclinical models through this alternative pathway that is not so much related or connected with the gonadotropic pathway. Just to finish up, I would like to acknowledge the important contribution of the members of my team, especially Juan Roa, Juan Manuel Castellano, María Jesús Vázquez, and Violeta Eras, who have actively contributed to the data that I have shown you today, our different collaborators in this particular project, and also our funding agencies. Thank you very much for your attention.